We're going to start with the endomembrane system. The endomembrane system contains the nuclear envelope. Remember, the nuclear envelope has nuclear pores. There's nuclear pores. Which allow for the mRNA to come out. It also allows for nutritional material to go in, amino acids, different types of amino acids and proteins to go in. It functions as a channel. These are the components of the nuclear membrane system. It's also the pathway for production of proteins. It's going to go from nucleus. This is something you need to remember because you will have a question on this on the test. To the rough ER. Or ribosomes. From there, it goes to the Golgi Apparatus. From the Golgi Apparatus, it makes a vesicle, and then it can leave the system by fusing to the plasma membrane. The endoplasmic reticulum is basically, if you go back, let me just go back to this picture so it's easier to see that. The endoplasmic reticulum is continuous with the nuclear membrane. The little dots that you see here are ribosomes. The endoplasmic reticulum has two different types, the rough ER and the smooth ER. The difference between rough ER and smooth ER is that the rough ER has ribosomes on it. The smooth ER does not have ribosomes. Depending on the functionality of the cell, you will have either more a rough ER or less rough ER. Cells that produce a lot of proteins are going to have a ton of rough ER. Cells that do a lot of detoxification, production of lipid modification, and all of that will have a large number of smooth ER. Continuous with that, you see this almost pancake-like structure, flattened stack of pancakes kind of structure, and that is Golgi Apparatus. The Golgi Apparatus has two faces. This face is the trans face. And this is the cis face. As you can see, this one right here is the formation of a vesicle. A vesicle is being formed. It's just the Golgi apparatus like pinches off a part of it and a vesicle is formed. Let me go back to the slides. The rough ER and the smooth ER. This is a good picture of it, so you can see where the nuclear pores are right there. Things, if you have an mRNA, the mRNA is just leaving the system, getting here, the ribosomes attached to it right here, and transcription and translation starts. One of the main functions of the smooth ER is to synthesize lipids. There are different types of lipids that your body needs, that your body produces. For example, there are different types of steroids. Cholesterol is a steroid molecule. A steroid molecule, just to recap from our previous from the previous chapter, steroid molecules are 4 fused carbon rings. These are all synthesized by the smooth ER. They metabolize. The word metabolize means metabolism. The word metabolism is a combination of anabolism and catabolism. Catabolism is to break something down. Anabolism is to make something. And so they do that with carbohydrates. The units for the monomers for carbohydrates, like glucose, is your body gets that through digestion. You're eating complex carbohydrates, whether it is glucose, sucrose, whether it is sucrose, maltose, fruit, or what is the other thing? Maltose, sucrose, lactose. You're eating either one of those, or even starches and glycogen and stuff like that. It goes through the process of digestion. In the process of digestion, it is broken down into its monomers. Then those monomers have to be remade into things that your body needs, and that is done by the smooth ER. Detoxification of all sorts of poisons is done by the smooth ER. So cells like organs like the liver and the kidney will have large number of smooth ER in them. They also play a vital role in storing calcium. Calcium is extremely important, not just for within your body, not just for your bones. It is also extremely important for your heart to beat. The electrical charge that you get in your heart, for your heart to actually beat, is done by calcium plus 2 ions. One of the most important tasks of the rough ER is to produce proteins. They secrete glycoproteins that are required for the system. Glyco coming from the word carbohydrates, proteins coming from the word proteins. So there are where proteins are covalently bonded to carbohydrates. These glycoproteins have multiple functions in the body. One of the things we looked at was on top of the cell, they also act as receptor proteins. They act as proteins that allow the body to identify different types of cells. They produce transport vesicles. Transport vesicles, anytime you see the word vesicle, all it is that a part of the plasma membrane pinches off and forms like a it comes from the word vessel or a vehicle, and then this one, it has a tag of the protein sticking outwards, and then it can be transported from point A to point B. This is one of the reasons why everything in the cell is made up of phospholipid bilayer. So you can pinch off a part from one area, and then it can basically fuse in another area, and then pinch off from a part of the Golgi apparatus, or the rough ER can pinch off, form a vesicle, fuse with the plasma membrane. A part of the plasma membrane can pinch off and fuse with the rough ER or the Golgi apparatus. This way, the net amount of membrane stays the same, even though parts of the membrane are going from one organelle to another. This is it is the membrane factory of the cell. This is where most of the membranes are produced. The next organelle in the system, in the endomembrane system, is the Golgi apparatus, also called as Golgi bodies. This is the best way to think about this is that it is like a massive shipping center, like the post office, where it receives the proteins that are produced. It kind of modifies the products, puts address. Think of it like putting address tags on a package. If you think of the proteins as a package, like a box of things that needs to be mailed, the job of the Golgi apparatus is to actually put an address tag on it. Basically puts a glycoprotein, sometimes a lipoprotein tag on it. It kind of sorts all the materials, figures out exactly what is the destination of which type of protein, and then packages them here packaging basically means putting address tags on it. These address tags are made up of glycoproteins. And then puts them into transport vesicles, and then these transport vesicles basically are transported to wherever they need to go. The cis face is the receiving face from where it receives materials. This is where it receives the vesicles from various organelles in the body, in the cell. And then the trans face is where it makes the vesicles and sends them out. The trans face is the shipping face. The next part of this is lysosomes. Lysosomes are very simple hydrolytic, containing hydrolytic enzymes that can break things down. They have a pH of 4.5. So it's using water to break down proteins. When proteins need to be broken down into their monomers, or polysaccharides need to be broken down into those monomers, it is the lysosome's job. It's a very, very simple bilayered organelle that has a very low pH. This plays a very important part in your non-specific immune system also. When the body finds something that's a foreign material, it is brought into the system, and then the enzymes basically digest it. Enzymes in the lysosome basically digest it. Let's look at. So let's say you have some sort of a bacterial infection. Your immune system has killed the bacteria, but the bacteria still has a lot of amino acids and proteins and other things that the body can use. It also needs to be broken down into their individual components, so it's not used up by other bacteria in the area. So a membrane is formed around this foreign material, and it's considered, think of it as this acts now as food for your cell. Through the process of phagocytosis, it creates like a vacuole. A vacuole is just an empty container that is an empty vesicle that contains food. If it's phagocytosis, it is food. If it's pinocytosis, it's water, liquid. This food vesicle then fuses, which the food is brought towards the plasma membrane. The plasma membrane then fuses it, puts it into a vacuole. That vacuole then goes ahead and joins with the lysosome to make a lyso complex. Forms a phagolysocomplex, and this phagolysocomplex then digests the food in there, breaks it into its monomers, and the body can use it. Lysosomes are also a big part of the process of apoptosis. Apoptosis is also autophagy. Let's assume that the cell has been infected by something and needs to be destroyed. If the cell needs to be destroyed, the lysosome basically opens up, and as soon as the lysosome opens up, it basically then destroys, because its pH is lower than what the cell is used to, it destroys the cell. This usually happens when the cell is infected by different type of viral particles, or during in vitro growth, when the body is laying an organizational plan, and for example, the webs between your fingers and stuff like that. When you were in vitro you had a skin fascia kind of thing growing between your fingers, and those need to be killed off, and this is done by the lysosomes. Basically, it's apoptosis. The cell is killing itself. This is a good image of a phagolyso complex. That would be a phagolyso complex, where the food, the phagos cell is combining with the lysosomes to break down, and then the food particles are then broken down, because it's basically the same reaction of polymer plus water gives you monomers, and then it breaks down into its monomers, and if these monomers are something that the body can use, it does go ahead and use it. Plant cells have more than one vacuole. There are major vacuoles. There's a central vacuole that basically even gives the plant cell its structure. Food vacuoles are formed through phagocytosis. And in the case of, if it's a liquid, it's a pinocytosis. Contractile vacuoles are usually seen in freshwater protists. Protists are single-celled eukaryotes. They're seen in protists. Inside the cell, there needs to be homeostasis. There needs to be a certain amount of water. There needs to be certain amount of the solution has to have a particular ratio of solute to solvent concentration. However, the water outside may or may not be of the same concentration as inside the cell. So if you have, let's say, let's just do this. Let's say outside you had, if this was a cell, and this one has a concentration of 20% of solute concentration, and it was in water, and that water was, let's say, 40% solute, that means there's less concentration of water on the outside and a high concentration of water on the inside. There's more water here. There's about 80 units of water here, and there's about 60 of water here, right? Diffusion, the concept of diffusion is when things move from a high concentration to a low concentration. Diffusion is when things move from a very high concentration to a low concentration. If you spray perfume on one side of the dome, over a period of time, you will smell it on the other side. So wherever you sprayed the perfume, you had a very high concentration, and on the other side, you had a low concentration, and the molecules move from a high concentration to a low concentration. When you have a cell submerged in water, and if this one is 80% water and 60%, the water is going to start going outwards, right? And it's going to keep doing that till the net movement is zero. On the other hand, if you had another cell where the concentration of water was greater on the outside, let's assume there was 90% H2O here, then the water will start going inwards. If the water starts coming out of the cell gets dehydrated. If the water starts going into the cell, after a period of time, it can burst. So contractile vacuoles, the job of the contractile vacuoles is to make sure that if water is entering in or the ions are entering in, they're pumped out in order to keep this balance, so that this balance is not disrupted. The cell needs to keep internal balance. It cannot keep disrupting the internal balance. Contractile vacuoles' job is to make sure that when these protists that are living in fresh water, if the concentration of water on the outside changes, it's able to maintain the concentration of water inside by pumping anything excess outside. Central vacuoles are found in plants. They hold many organic compounds as well as water. You have seen the function of central vacuoles in your daily life. If you take a fruit or vegetable or anything and leave it on the counter over a period of time, it kind of dehydrates. The reason for it to dehydrate is that that your fruit or vegetable is actually using up, is losing some water to the outside because of the a difference in concentration. It's also using up some of the water to maintain itself. If you leave a fruit outside, it gets slightly dehydrated, and once it gets dehydrated, it gets much sweeter. The reason is because the central vacuole contains sugars as well as water. So as the concentration of water goes down, sugar gets more concentrated. The job of the central vacuole in the cell is also to provide, it kind of pushes up against the cell membrane and kind of gives it shape and provides it something called as turgor pressure. So it keeps the shape of the cell because it's filled with sugar as well as water. This is a really good picture for you guys to understand the pathway which it takes. It comes from the nuclear pore. Let's look at mRNA. So we can take mRNA as an example and then build it from there. The mRNA comes out through the nuclear pore and then is translated in the rough, transcribed and translated on the rough ER. The ribosomes do that. From the rough ER, then it makes a vesicle, and it's received by the cis face of the Golgi apparatus. It's modified by the Golgi apparatus. It puts in the required tags that it needs to put in. That's like the address tags, which are just glycoprotein tags that it puts in, and then creates a vesicle. Then the vesicle basically fuses off with the plasma membrane. Something you have to remember here is this cannot be a one-way channel. If it is a one-way channel, then eventually what will happen is every piece of the ER would have pinched off, attached itself to the Golgi apparatus, and then every part of the Golgi apparatus would have pinched off, attached itself to the plasma membrane. The plasma membrane size would just keep increasing, and every other organelle would be decreasing. That is not how this takes place. So while there's this is a channel for things to go out, the channel for things to go in would be comes to the plasma membrane, pinches off from the plasma membrane, goes to the Golgi, and from the Golgi it again forms a vesicle and fuses wherever it needs to go to. It is a two-way process that maintains the amount of phospholipid within the system. More phospholipids are produced, but this is one of the ways it is. We're going to look at, so that is the endomembrane system. The next part of this is we're going to look at chloroplasts, mitochondria, and perioxosomes. Chloroplasts and mitochondria are found, both chloroplasts and mitochondria are found in plant cells. However, only mitochondria is found in animal cells. Plant cells do contain mitochondria. Please remember that, because plant cells also need ATP. The essential function of mitochondria is to produce ATP. Its job is to take C6H12O6, add oxygen to it, hence the word respiration, and break it down into CO2 plus H2O. In the process of this, depending on the type of respiration it is doing, it's going to produce anywhere from 36 to 38 ATP. In the case of chloroplasts, it's the site where photosynthesis takes place. That is the opposite of this reaction. It is taking CO2, adding H2O, using light to make C6H12O6 plus O2. This process of photosynthesis is only done by plants, certain bacteria, the animal prokaryotes. In the category of prokaryotes, bacteria, there are certain types of bacteria that do it. Protists do it, but they all have either a chloroplast or a chloroplast-like membrane. The process of cellular respiration is pretty much universal. It's done by almost every organism that there is. That it is done by every organism that we know of, because ATP is the currency of energy for every organism. Perioxosomes' job is to do oxidation. In the process of cellular respiration, it produces an H2O2 is produced. So you're going to require that to be broken down and made into H2O. And that's what this is. This one is doing. Mitochondria is not a part of the endomembrane system. Mitochondria and chloroplasts are both a part of the endosymbiotic theory of evolution. They have a double membrane. They make their own proteins. A certain number of proteins they make for their own by the ribosomes that they have. They also contain their own DNA, just like prokaryotic cells that have a circular DNA. Chloroplasts and mitochondria both have circular DNA. They're essentially like a prokaryotic cell within your eukaryotic cell. However, they don't qualify as a cell by itself because they're not capable of doing all the other functions that a cell does. They're not capable of reproduction. They're not capable of living independently by themselves outside the cell. So while they are able to produce their own proteins, have their own DNA, you might have heard of mitochondrial DNA in terms of your maternal DNA is your mitochondrial DNA is all from your maternal line, because all the organelles are in the cell come from the egg and not the sperm. So the mitochondria, you share the exact same, with due to mutations there'd be some differences as with your mother and your mother with her mother. So the line of mitochondrial DNA is used to understand evolutionary lines better. The mitochondrial DNA is basically found, mitochondria is found in nearly all eukaryotic cells. In prokaryotic cells, there is a mitochondrial membrane. There's a membrane that does cellular respiration. Let's look at a picture of this and it'll be a lot easier. They have a double membrane, just like everything else. There is a bilayer. The inner membrane is kind of folded inwards, and these are called cristae. It's on these folds where cellular respiration takes place. A large part of the cellular respiration takes place in the inner membrane, on the membrane itself. They have a ton of free ribosomes. These free ribosomes produce the proteins that that the mitochondria requires. They have a DNA. The DNA is a circular DNA, so they are able to produce their own mRNA to allow for the transcription and translation of proteins. They also have cytoplasma like solution in there, and the cytoplasma of the mitochondria is called the matrix. The next member of the endosymbiotic family is chloroplasts. Chloroplasts belongs to the family of organelles that are called plastids. Plastids are organelles that are involved in either storage of food or making of food. They contain the chloroplasts contain a green pigment called as chlorophyll. The job of this chlorophyll is to actually absorb light and break water, and we'll do this more in detail when we do photosynthesis. Chloroplasts are usually found in all green, everything that is green, whether it is leaves, in algae, in single-celled protists that are green. All of them contain that. Let's also look at a picture here. They have these disc-like stacks called as thylakoid. A stack of thylakoids are called granum, and multiple granum are called grana. They also have their own ribosomes, just like the cell has cytoplasma and the mitochondria has a matrix. They have a viscous liquid-like solution inside the cell, and that's called a stroma. They also have a circular DNA, hence they're able to make their own mRNA and then are able to produce their own proteins using the ribosomes that they contain, the free ribosomes that they have. The next organelle we're going to talk about is the perioxosomes. The perioxosomes are single membrane. They're not a bilayer. So every organelle that we talked about so far has a bilayer. They have a phospholipid bilayer. In this case, perioxosomes have one layer. Perioxosomes are involved in the process of oxidation. Oxidation is loss of electrons. The oxidation does not necessarily involve oxygen. Originally, the term was used when oxygen caused the electron loss in reaction. So one of the ways I remember this is oil, which is oxidation is loss, reduction is gain. So this is the way. It's just a mnemonic to help you guys remember it. It'll keep coming up multiple times as we go along. So it essentially does this process. H2O breaks down to form H2O plus 2, and then you can balance the reaction. Oxygen is then used to break down other different types of molecules. This is a good picture of all three of them. The next large class of organelles that we're going to look at is cytoskeleton. As the word suggests, cyto meaning cell, skeleton is just a network of fibers that gives structure to the cytoplasma. It not just gives structure, it allows for movement of organelles. It kind of lays down these railroad tracks kind of things on which organelles, the vesicles can travel from point A to point B. It allows for the cytoplasmic streaming to happen. It allows the organelles to anchor from one place to in one location. It also allows the cell to anchor. It is a part of moving the cell from point A to point B. In the beginning, we talked about cilia and we talked about flagella, and all of these are a part of the cytoskeleton. The three molecular components of this are microtubules, microfilaments, and intermediary filaments, and we're going to look at each one of them separately. This is what I meant by when they said like monorails. It kind of lays these railroad tracks. So let's look at microtubules as a piece. Microtubules are one of the most common type of part of the cytoskeleton. They are hollow tubes that are laid in with the monomers are called tubulins. There are alpha tubulins and beta tubulins depending on how they're structured. They basically will form a structure where the tubulins kind of lay themselves that way, and they form like an alpha helix structure. So they're going in this direction, and it's a hollow tube. Their biggest job that they do is, besides maintaining shape, they are vital in laying those monorails for organelles for vesicles to go from point A to point B. They are also vital in cell division. When your cell has to divide, the chromosomes have to move. If you guys remember from high school biology, mitosis and meiosis. In the process of mitosis and meiosis, microtubules are going to be extremely important. They allow the migration of the replicated DNA to the two different poles, and then allow for the cell to break up into two parts, two pieces. The parts that here you have to remember very carefully is that they are made up of tubulins. Tubulins are the subunits. So when the track is being laid, tubulins are just laid ahead of it, and then the vesicle can move along the tubulin. And when it needs to be disrupted, all it has to do is hydrolyzes the tubulin subunits from the back end. So if it's laying tracks, it just goes ahead and lays a bunch of these. Move, sweetie. It kind of lays these tracks where it's forming. It uses it's polymerizing on this end, and then when it needs to break it up, it just hydrolyzes it. So it adds water, and then these the peptide bonds are broken down, and then it just breaks it up. So it's not like once the tracks are laid, they're not just there. The tracks are laid when needed and removed when needed. The next type of filament is the microfilaments. These are a big portion, a big function of this is to do muscle contractions. When you contract your muscles, it is a function of the actin and myosin proteins working in them. The fibers are made out of actin, and then you have the myosin protein going back and forth on it. Another huge thing that they do is cytoplasmic streaming. Cytoplasmic streaming is also called protoplasmic streaming. Sometimes it's called cyclosis. This one is basically the flow of the cytoplasma within the cell itself that then facilitates the speed with which molecules are transported from point A to point B. Organelles are moved around the cell. All of this is done that. It plays a vital role in cell motility. The cell itself moving. For example, if you look at amoeba, which is a single cell eukaryote, the way the single cell eukaryote moves, if there was a piece of food right here and there was an amoeba right there, and this was an amoeba cell right there, what it does is that it kind of creates these pseudopods. It extends its cell outwards to engulf the cell. It creates false legs, pseudopods. It creates that and engulfs it, and that is a part of microfilaments jobs. It is also involved in cell division. In the cell division itself, the microtubules is involved in the division of chromosomes, whereas microfilaments are involved in cell division itself. The next one we're going to look at in this chapter is intermediary filaments. Intermediary filaments they're a little thicker. They're thick fibrous cables that are laid there. Their job is essentially to provide shape to the cell. They allow for the nucleus and other organelles to anchor themselves. They've also formed something called as a nuclear lamina, and we talked about it when we talked about the nucleus. They're super critical for the nucleus to provide it a certain shape. It regulates all the various nuclear processes that are taking place, including transcription, translation, and replication. They are a part of the keratin and the keratin family. The keratin family is the one that makes nails and hair.